So stage is all yours, Rajesh and Andrew.
Hello, everyone. Welcome back. I hope we are on now.
Yes, you are. Okay, continue.
Yeah, thanks, Andrew, for the lectures today. We have a few questions, and I will read them out one after another. Every now and then, we'll also look at the Q&A to see if somebody has posted a question there, so that we can make it a little more interactive. So the first question is from lecture 5, and it's the following. Proteins can be synthesized from amino acids. Can we synthesize a protein from another protein? Please comment on it. What are the conditions that are required for proteins to become stable? Can we read and write proteins like DNA? So that's the question.
Okay, so there are several questions here. First off, can we synthesize a protein from another protein? In principle, yes. So as you recall from the lecture, actually, let me share my screen and bring up the notes for the lecture. Lecture 1. So proteins are defined by the peptide bond, and in any protein, you have a chain of peptide bonds forming a polymer such as this. So at each end, there will be an amine end and an acid end. So in principle, there's no reason why you can't combine proteins end to end in order to create new proteins. This actually, as far as I know, this does not happen so much in biological systems. There is an active area of research called protein-protein interaction, but it usually does not involve the combination of proteins by reaction. It's usually interactions between proteins by electrostatic forces. The next part of the question was, what are the conditions required for proteins to make them stable? I don't have specialist knowledge about that, but I think it depends on what kind of protein you have. But generally speaking, it's what you would expect. Some proteins are sensitive to temperature, some are sensitive to acid, some are sensitive to all kinds of other things that can potentially attack them, and others are very stable. So there's a very wide range of conditions that will support proteins. Can we read and write proteins like DNA? So I can see where you're going with this. So in a protein such as this, let me expand this a little bit. We have these R blocks. So these represent individual amino acids, and I guess the question would be, given a sequence of amino acids, can you just read them off like we can read DNA? The answer is sort of. So DNA sequencing is very famous. We know that if we're given a molecule of DNA or RNA, it is possible to look at it, inspect it, and directly read the bases from the DNA or RNA. In proteins, it's not that easy. So while there is a sequence and while there is a science of taking proteins and trying to read their structure by trying to identify the amino acids, it's not quite so easy. The standard technique is mass spectrometry, in which case what you do is you break the protein into small chunks, and about all you're able to determine is the mass to charge ratio of those chunks. And from that, you have to deduce the structure of the protein. There are other methods, such as I think X-ray crystallography is used for this. So there are other methods to attempt to determine the structure of a protein, but it's not as simple as sequencing. The general science of trying to understand the structure of a protein is called proteomics, and there's a rich literature on it, and there's some interesting signal processing problems. So that I think that summarizes my answer to that question.
The next question is, can DNA genetic information be compared in some sense to a conventional binary coding scheme?
Okay, this is a very interesting question. So DNA is basically digital information. It's not binary. It's 4 array, so there are 4 bases. I'm not terribly familiar with this, but I am aware that there are there are error correcting capabilities in DNA. It is possible for enzymes that read and transcribe the DNA to understand when an error occurs and to potentially correct it. So there are ways in which that can happen. It's not sophisticated in the sense that DNA is, for example, not implementing an LDPC code, but there are certain error correction mechanisms that have evolved to use DNA.
May I have an add-on question to this?
Certainly.
Is there a evolutionary biological basis for for this forary rather than a binary or a ternary code?
Good.
Some thoughts on that?
So this is pure speculation. I don't know. I suspect that. So at minimum, you would need 2 bases, I think, to express anything. So by the hybridization structure of DNA, so between the two helixes, the base is hybridized. So on the one side you have a base, and on the other side you have the complementary base. So this implies that however many nucleobases you have, it should be even, because for each base there must be a complementary base. I expect that, so it is, you can easily imagine we have 4, so it was potentially possible for 2 to arise. Why don't we use 2? Perhaps because the structure of, or the complexity of organisms that you can develop using 2 is too low, or perhaps the enzymes for translation become too complicated because they have to look over now, I guess they'd have to look over many more nucleobases than with a 4-ary scheme. As far as 6 or 8 or more than that, again, this is pure speculation, but perhaps chemically there don't exist such bases. Like you don't have 6 bases or 8 bases with the same properties as the 4 that we have.
Next question is, if adenine always pairs with thymine, can a sequence with ATCG be self-paired in the same DNA?
Yes. So this is actually a big problem. And this is actually part of error correction in DNA. So some of the correction is simply the transcription, and other parts of the correction are preventing things like this from happening. So what you can have is, in transcription of for example, RNA, you have a single strand, and what can happen is it can bend and hybridize with itself, and this causes significant problems. So that's definitely possible. There are mechanisms to avoid that.
Okay, so I think those were the questions from lecture 5. Let me check if there are some questions on the Q&A. There are none. Okay. So
I
maybe
think
let's
there's
go to
one more. I have this one more question from lecture 5.
Lecture 5, yes. What is the significance of DNA transcription and translation in molecular communication?
So that's an excellent question. In the series of lectures that I've prepared, I don't touch on this very much. I simply introduce transcription and translation to talk about where proteins come from, and then we talk more about protein dynamics. But there is an emerging field of study in DNA data storage. So DNA transcription and translation is very important for DNA data storage. And basically what you do there is you take DNA and you use it to encode whatever information you like, as you might write a file to a hard disk drive. So this has excellent properties in terms of stability, in terms of longevity, and in terms of energy, in terms of density. So there are actually many both research groups and companies trying to do research on recording of data on DNA and retrieving that data from DNA. So you can easily imagine that what you could do to send a message is you could record it on DNA, package that DNA in a vesicle or something, and then emit that vesicle to a receiver.
Okay, we'll go to lecture 6 questions now. There's one question which asks for an example of a macro scale molecular communication. So would you like to?
Right. So looking forward to the final lecture, lecture 16, you will see many examples. So in the lab, we have designed many systems that actually implement molecular communication at the macro scale. I also go through some potential applications of it, such as maintenance of sewer systems or search and rescue and so on. So I'll leave that to you to look at. But if we think about natural examples of macro scale communication, animals communicate using pheromones. That's an example of macro scale molecular communication. You emit a scent, and other organisms of your same species or other species detect that scent, and they receive some sort of message based on that.
I have a sort of slightly related but unrelated question. Is there an example of a computing engine using molecular systems that you can suggest or give an example? You could say that the protein folding is a minimum energy configuration, so you could say it's computing a minimum. But is there something more concrete that you could suggest, perhaps?
So this is an excellent question. So if we're talking in terms of computing, about 20 years ago, there was a lot of research in a field called DNA computing. So what was realized was that DNA hybridization could carry out simple computing tasks. There was a lot of interest in it because someone proved that this technique was Turing complete. In other words, it could compute if the DNA was structured in the right way, it could compute any function, any function that a computer could compute. And the fact that there are potentially billions of DNA molecules in a small space, this meant that the computation was highly parallelizable. So in other words, the computation could occur very, very quickly. The practical difficulties of DNA computation meant that it didn't become practically interesting. So I don't think there's any actual implementations of this. But more recently, we've seen interest in ubiquitous computing, where any kind of potential computing substrate could be used. Neuromorphic computing, which can use neural principles, and so you can imagine that these ideas will come back, and we can think about doing computations in chemistry. So this is definitely an area of active and increasing research interest.
Next question is a more direct question related to lecture 6, and it might be related to how in property 1 the Gaussian distribution is with zero mean.
Right. So
I hope you are getting the particular context where property 1 is described. Might be with
yes, I think the question asker is referring to this part of the notes. So here we have basically this is the property of independent increments. So if I have a Brownian motion at a certain time, the change at a later time from an earlier time is distributed with the Gaussian distribution with zero mean and variance some sigma squared, some constant times the difference in time. Why is it zero mean? So if you imagine the Brownian motion starting at the origin at time 0, we know that it's zero mean. So it'll spread out from there. The distribution of potential positions will spread out from there by the Gaussian distribution. So if I fix it at time t1 at a certain position and I ask how it evolves from that position, it is again zero mean. So in other words, I fix it at bt1 and ask later on at bt2 how will it evolve, it will actually just spread out from bt1. So the difference between bt2 and bt1 is again zero mean. So this is something like the Markov property. In fact, what we could do is we could consider this as what's the probability of BT2 given BT1. In that case, the mean of the distribution would be BT1. The difference between the two, the mean is 0. I hope that's clear.
Next question is related to the Markov property. Is it valid for the 3D Brownian motion as well?
Yes, it is valid for 3D Brownian motion because each dimension of the Brownian motion is independent. So because it is the Markov property is valid in each dimension, it is valid in all the dimensions.
Here I have a related question, perhaps an advanced one. So Brownian motion in one dimension is recurrent. In 2 dimensions, it doesn't hit points, but it hits sets. Any set of positive measure, you know that it will hit with probability 1. But in 3 dimensions, we don't have this property. And molecular communication happens in 3 dimensions. What are the consequences? Have you compiled a list of problems associated with this, and can you share it with us?
Yes, so we talk about in this lecture we mostly talk about 1 dimension because it's excuse me, in this course we normally talk about 1 dimension because it's mathematically straightforward. In 3 dimensions, you can you can use the property that I just said, which is that each dimension of the motion is independent. In 3 dimensions, if the receiver is an infinite plane, then only the distance to the receiver is important. So this again becomes a 1-dimensional system. If on the other hand, if the receiver is some ball or something that is not infinite, then you're right. The molecule might miss the receiver. So this is something when you design a molecular communication system, you have to take into account that a lot of your signals will not arrive at the receiver. But I mean, this isn't so different from wireless communications. Almost all of the energy in an isotropic antenna will be directed in useless directions, and only some of it will arrive at the receiver. Unfortunately, there are not very many closed form solutions for things like first arrival time, and most of them only exist for things like the one-dimensional case. So that's another reason why we focus on 1 dimension in this course.
All right, thank you. The next question is about can the diffusion coefficient be varying at different locations if the medium has uneven temperature, uneven viscosity?
Definitely. So we, again, for convenience and simplicity, we normally assume in this course that the diffusion coefficient is the same everywhere and that the medium is homogeneous. But there are definitely real-world examples of the problems caused by uneven temperature and viscosity. So this would cause uneven diffusion, and this is potentially something that you'd have to take into account in designing a real molecular communication system. But again, in this course, because that's a difficult issue and doesn't lend itself to clean closed-form solutions, we tend not to deal with that in this course. But it is an issue. I also see that there's a question in the Q&A.
Maybe if Adarsh Patel could be brought to the stage, Adarsh could ask the question by himself. Could one of the volunteers bring him to the stage? Adarsh, are you on stage?
Not yet. 1 minute. Oh, disallowed? 1 minute, sorry. Yes, he is on stage.
Adarsh, please go ahead and ask your question.
Yes, yeah. Hello, Andrew. First of all, thank you for this lecture. I mean, it's a very good lecture series. It's I guess not only for me, for others as well. It's very explanatory, and it's very helpful for everyone. So now my question is, in the non-absorbing receiver, the molecule continues its Brownian motion. Okay, so it can come inside the region, inside the receiver region, and then it can very well move away. And this is for a single dimensional case, and if we consider the similar scenario for multiple dimensions as well, because I'm assuming that the case will not be limited to 1D case, because I guess people have developed some 2D models as well. So for in that case as well, how is this thing encountered?
Good question. So with non-absorbing receivers are actually very difficult to analyze. So from the level of individual molecules, you're exactly right that if you observe a certain number of molecules inside the region, that count has time dependence in a complicated way, because a molecule can leave and then come back very quickly. And be counted in the same way as a molecule that has been propagating for a long time that arrives for the first time. The normal way in which we analyze non-absorbing receivers is to actually, in this lecture series, we do talk about counting those molecules, but we actually don't tend to deal with direct counts in non-absorbing receivers. We tend to deal with distributions, we tend to deal with concentrations. And another thing that we tend to have to do is we tend to have to assume independence. So I actually don't have as much experience with non-absorbing receivers as with absorbing receivers. I do know that it's much more difficult to deal with. We do talk about some techniques in this lecture series. I can't remember exactly in which lecture, maybe lecture 8. We do talk about some techniques for dealing with these kinds of situations, but it is much more difficult.
Thank you, Adarsh. Thank you, Andy. Yeah, we'll now go back to the lecture-related questions that we got beforehand. Lecture 7 now. Can we model these types of receivers for macro-scale communication? So sort of related to the first question in lecture 6, but more of a modeling question.
Yes. So in macro-scale communication, the model is more like the non-absorbing receiver. So what we found, in fact, we discovered this when, sorry, is there a...
Yeah, sorry, I think either I had a poor connection or maybe you did, but you're back. Please go ahead.
Okay. So as I was saying, we can model these types of receivers using macro-scale communication. The best model to use is the non-absorbing receiver. There are some techniques that are laid out, I think, in a later lecture. What we've discovered is that through experimentation, what we discovered is that because the molecules aren't absorbed, they tend to linger in the region of the receiver, and that causes a very long tail to the impulse response of the channel, and the ISI will tend to be significant unless you use something like drift, like a big fan, to blow it away. So you can definitely model macro-scale communication using the techniques that we're talking about in this course, and I think in a later lecture there will be some examples provided.
The next question is related to drift. In the presence of drift, the transmitters and receivers may themselves get affected and change their position. How does it affect modeling? Any thoughts on that?
Yeah, this is an excellent question. So it's a complicated issue. Normally in this lecture series, we will assume the transmitters and receivers are fixed in fixed positions, simply, again, because that's an assumption that can be made for convenience. But there is a very active area of molecular communication research called mobile molecular communication, in which the transmitters and receivers also propagate by Brownian motion. So there have been several papers recently, actually within the past year or so. This has been a significant area of research, and is very practical too, because you would assume that in a nanomedical application, these nanomachines that are floating around your bloodstream would also have to move with the blood. How it affects modeling? So it affects everything. Basically, what you would have to look at is the relative motion between the transmitters, the receivers, and the signal-bearing molecules. Sometimes it works out nicely. So for instance, basically what you'd have is linear combinations of the motion, or you would have to translate the motion into one frame of reference. Sometimes that works out very nicely, depending on the assumptions you make. Sometimes it's more complicated.
Next question is about nomenclature. Why is the distribution called inverse Gaussian?
Okay, so the way it was explained to me, and I'm not sure if this is true, so it's definitely not 1 over the Gaussian distribution. So it's not like Gaussian to the -1. Instead, it's better understood as an inverse problem. So the Gaussian distribution for the Brownian motion, as we see here, is the distribution of position as a function of time. But the inverse Gaussian distribution gives the distribution of time as a function of position. So it's basically an inverse problem is the way to interpret it.
Next question is, could you please provide some examples of absorbing and non-absorbing types of receivers that can be made or that exist in nature?
So this is a really nice question. So we're going to talk in this lecture series about signal transduction. We've already talked about it a little bit, but signal transduction receivers are basically absorbing. So these are receptors that exist on the surface of a cell. A molecule called a ligand will attach to that receptor, so it's absorbed by that receptor, and then it's processed, so it's turned into what are called second messengers. So basically, it's then taken inside the cell, and something else is done with it. So this is an example of an absorbing receiver in nature. Non-absorbing types of receivers, actually, I'm not terribly familiar with those that might exist in nature. One example that I thought of was a calcium channel. So again, on the surface of cells, I think we've talked about this before, you have ion channels that can admit external molecules inside, and often what they do is they allow ion currents to pass from the outside to the inside of the cell. So you could imagine a concentration of ions in the surrounding environment. The channels open, and those ions then enter the cell and cause some internal change. Sorry, excuse me.
Yeah, let me see if there are any other questions on the chat. Okay, I don't see any. So let's go to lecture 8 and the questions we received beforehand. So it's related to the question that Adarsh also asked. So in non-absorbing receivers, can the same molecule be counted multiple times? So maybe we can hear you respond to that once again.
Yeah, the answer is definitely yes, and that causes huge problems for the analysis. And in particular, if you imagine, if you can think about counting one molecule or counting the number of times a molecule arrives, imagine that molecule at the boundary. So if the molecule is at the boundary and you're interested in counting the number of times that molecule arrives, then the number of times is actually very large. So it can oscillate basically at the boundary between inside and outside of the region. So there are many complications for non-absorbing receivers that make them difficult to analyze in the same way as absorbing receivers.
I think the next question is exactly the same about what problems it create to the receivers. Would you want to respond, or we can skip the question?
I think we've gone over this in some detail, but just to emphasize, non-absorbing. The reason why absorbing receivers are very nice and clean and simple to analyze is because of the Markov property. So once you observe a molecule at the boundary, you have all of the information you need about its prior propagation. The trouble with non-absorbing receivers is that you can't tell when you've seen them previously. So if, for example, once the molecule arrives, you could tag it and you could say, I have seen this molecule before. You gain nothing because of the Markov property, you gain nothing by observing it again. So in fact, you could ignore all of those molecules if you like. So like I said, the difficulty comes with the fact that you don't know which ones of these molecules you haven't seen before.
In that context, again, a somewhat technical and perhaps advanced question. The modeling by Brownian motion causes particular technical difficulties because it will cross the barrier or the receiver barrier or boundary infinitely often.
Yes.
And there are these local time issues associated. One can characterize those things, but it goes up and down infinitely often. From a communication perspective, these are issues.
Yes.
So what might, in practice, do we just focus on building absorbing receivers and not worry about this? Is that the way one would go about handling the experiment?
It's a very good question. So I would answer that in two ways. So the way in which I've structured my research is, on the one hand, I look at theoretical limits. So I'm interested in what's the ultimate performance that you can potentially get from a molecular communication system. In that case, what we tend to do is we tend to look at the cleanest, most mathematically tractable possible situations. So that's why I tend to emphasize absorbing receivers in this lecture series, is because they tend to lend themselves to the cleanest and simplest interpretations. And in fact, you can show there's a theorem that states that absorbing receivers have the highest possible capacity of any timing-based receiver.
Mm-hmm.
So if you're interested in ultimate limits, what you're interested in is absorbing receivers. On the other hand, in experimentation, sorry.
Yeah, could you clarify highest or lowest? The worst
Highest.
case or highest? So
They're the best case, and it's for exactly the same reason that I just mentioned. If you tag if you have a non-absorbing receiver, but you can tag a molecule when it arrives, then it reduces to the absorbing receiver. If you throw out those tags, you throw out information, so you can't do better. So that's the basic reasoning. So it's a best case scenario as far as absorbing receivers are concerned. However, when you do experimentation, it turns out that non-absorbing models are most useful, and so you must deal with them when you consider actual macro scale implementation. So hopefully somewhere the two techniques will meet in the middle some point in the future, decades away. But for now, we are looking at theoretical limits with absorbing receivers, practical implementations with non-absorbing receivers.
The problem is mitigated to some extent when there is drift from the transmitter to the receiver. So I guess to some extent in real life, we could build systems with a marginal drift.
That's exactly,
I have another question, a related question. So even if you have absorbing receivers, because you do not tag the molecules in the real system, let's say it's still absorbing, but you still can't tag it, then there is out of order delivery. How do you handle that? It's viewed as noise, or what's the
what you do is you tear your hair out, because it's a very difficult problem. So some of my first papers had to do with out of order delivery of molecules, and there's a famous result actually by a pair of Indian mathematicians that even to write down the probability density function. So let's say I have a vector of times, let's call it x, at which I release molecules. I have a vector of times y at which I observe the molecules at the receiver. Even to write down the probability density function of y given x is it belongs to a computational complexity class that's worse than NP hard. So it basically requires you to sum over all of the possible permutations. So this is a very difficult problem. And I had done some research on it about a decade ago and gave up because it was too difficult. I have a colleague or a friend named Chris Rose, who has done some more recent research on it, has made some progress, but this is an open problem. And if a simplification could be found, I think it would be very interesting.
But what is the specific problem? You want to figure out the order in which it arrived? That you might not be able to tell. But which is the NP hard problem here?
It is basically because any molecule that arrives could have arisen from any before it, and there's a permutation in mapping from transmitters to receivers. So it turns out there's no simplification. You must account for every possible permutation, and there's no known way to simplify that.
So it's the decoding problem, maximum likelihood.
Correct. That's right. That's right.
Okay. So let's see if there are any other questions from the audience. Maybe if somebody would like to raise their hand and ask a question, we such questions.
I see there's a question that came up.
Yes. This is, I do not know who has, maybe it is Adarsh once again. I'll read it out. For large N and K, we invoke central limit theorem and say binomial goes to Gaussian. Are molecules independent of each other ideally? And if not, how does this analysis change? Would you like to respond to that?
Right. So I assume this is concerning the non-absorbing receiver. So what you normally do is you count the number of molecules that arrive in the region, in the transmitter, in the receiver region. This would be modeled at any given time instant. This would be modeled by, the binomial distribution. And as the number of molecules increases, then it becomes the Gaussian distribution. I think this is an argument that we go through in this class. So the issue is that if you take a certain observation right now, you assume that it's Gaussian. You look at that same observation later, there's actually certainly going to be a correlation between those. So what you can do, we don't go through this part in this series of lectures, but what you can do is you can either estimate or potentially in some circumstances exactly calculate what the correlation is between them, and then that becomes a jointly Gaussian process, which is relatively easy to deal with. So that's one possible approach you could take.
Okay. I think that brings us to the last of the questions that were asked, and it's also past time. So thanks very much, Andrew. I'll now maybe hand things over to Adrish.
Yeah, thank you, Andrew. Thank you, Rajesh. Very interesting talks, Andrew. And we'll see you again. We'll see you again tomorrow. Yeah.
Sounds good.
Okay, bye.
See you tomorrow.
So tomorrow, please check the schedule, detailed schedule, not program at a glance. Tomorrow morning at 6 a.m. Okay, because I don't know the start time yet, because I have not received the video. So I will, the moment I receive it, I will find out how much time the duration is. Accordingly, I will set the start time. So please, early morning when you get up, please check the detailed schedule to find out the start time for tomorrow. Okay, thank you.
Thank you very much.
Bye.
Bye, everyone.
Bye.
We'll meet.